due to the hyperplasia of both prostatic, stromal as well as epithelial components. And it leads to the formation of large, fairly discretely enlarged nodule in the periurethral region of the prostate. And how it looks on the gross examination. Here you can see its external surface, that is the multinodular in appearance, and these nodules are of variable size and shape. And this is the cut surface of it. It looks yellowish pink in color, and prostate is secrete the seminal fluid. So when we press it, from that the prostatic milky fluid is came out. And on microscopic examination, mainly the two features are seen. Here you can see glandular hyperplasia, and beneath that, in between, there is a stromal hyperplasia is seen. Now, what is seen in the glandular hyperplasia? The glands, they show the papillary infoldings. Here, papillary infoldings you can easily demonstrate. Okay. And in the lumen of gland, there is a amorphous material, or it is the hyaline material, and we called it as the corpora amylacea. And the stroma, it shows the fibromuscular hyperplasia, and there is a sometimes combination of both glandular and stromal hyperplasia. So these all about the hyaline changes. So up to this, we have seen the hydropic degeneration or the hydropic changes, then the hyaline changes. These are the two different morphological forms of the reversible cell injury. Now, third one is the mucoid changes. Now, mucus, all of you have learned the histology, and you have learned that the mucus is produced by the mucin secreting epithelial cells and also by some connective tissue cells. Now, the example of the mucoid changes are the catarrhal inflammation of the respiratory tract. In case of common cold, we also called it as a sinusitis. Then whenever there is an obstruction of the ducts, which leads to the formation of mucocele of the gallbladder or the appendix, and also it is seen in the mucin secreting tumor or the mucus secreting tumors, and that is the mucinous cyst adenoma of the ovary. Now we will see the morphological changes in the sinusitis. See here, whenever the sinusitis is developed, the respiratory mucosa, all of you know the respiratory mucosa, it's a ciliated columnar epithelium with presence of a goblet cells. So whenever there is a sinusitis is developed, this mucosa becomes swollen and it secretes the large amount of mucus by their goblet cells. And this subepithelial stroma or the submucosal stroma, it shows the edematous changes with the small blue and red dots that are the inflammatory infiltrates. So these are the mucoid changes which are seen in the respiratory mucosa in case of the sinusitis. Now the other example, that is the mucinous cyst adenoma of the ovary. So whenever the mucinous cyst adenoma of the ovary is developed, on external surface, we can see the smooth shiny surface with a basilated appearance and large cyst is seen. Whenever the mucinous cyst adenoma occurs, it is usually unilateral in nature, and when there is a serous cyst adenoma, it is bilateral in nature. And when we see the cut surface of it, it shows this multiloculated cyst, and when we cut this cyst, the viscous thick gelatinous material that we also called it as the mucoid material is came out. Now we will see the microscopy of this mucinous cyst adenoma. In mucinous cyst adenoma, the cyst lining epithelium are of two types. First is the endocervical type of epithelium, and in some mucinous cyst adenoma, intestinal type of epithelium is seen. Here the cyst wall, it is lined by the endocervical type of epithelium, that is the tall columnar cell with abundant intracellular mucin. And when there is an intestinal type of epithelium, it shows the foveolar-like epithelium and presence of a goblet cells which secrete the mucin. Now, if we want to demonstrate the mucin in the tissue, the various special stains are used, and that is the PAS stain. PAS stain will give the magenta color to the mucin if mucin is present in the cell. If we are going to do the special stain Alcian blue, it gives blue color to the mucin, and mucicaramine, it gives red color to the mucin. So you have to remember this spatial stain which we are used for the demonstration of mucin in the tissue, and that is the PAS, Alcian blue and mucicaramine stain. Now the last reversible morphological changes which is observed within the cell is the fatty changes. Whenever the neutral fat accumulated within the parenchymal cells, it leads to the fatty changes within the organ. The fatty changes most commonly seen in the different organs are the liver, heart, skeletal muscles and kidney. Today we are going to see the fatty changes in the liver as well as heart. It is the most commonly seen in the liver and heart. In all the organs, there is a clear vacuoles are seen due to accumulation of fat in the parenchymal organ, and it is seen in the form of a vacuole. So we have to differentiate it from the water or the polysaccharides. Okay, and for that we are going to do the spatial stain. So for demonstration of the fat within the tissue, the spatial stain is used, and that is the Sudan stain, oil redo and osmic acid. If we are do Sudan stain, then it gives black color if fat is present. Oil redo gives red color to the fat droplets, and osmic acid also gives red color to the fat droplets. Now we will see the changes are seen in the liver whenever there is a fatty degeneration occurs. When the fatty changes occurs in the liver, the weight of the liver is increased up to 4 to 6 kg. Their external surface, it becomes smooth and glistening. Its edges becomes rounded, and normal color of the liver is the purple. But when the fatty changes occurs, it becomes yellowish in color and soft in consistency. And when we do the cut section, the cut surface of it becomes bulge out. It is pale yellow to yellow in color, and it is greasy in touch. Here you can see the cut surface of the liver. When there is a fatty changes, it is yellowish in color. Now microscopic appearance. In the initial stages of the fatty degeneration, the centrilobular portion of the hepatocytes are affected. And initially, only the small fat droplets, they are accumulated within the hepatocytes, and we called it as the macrovesicular lipid droplets. But as the disease is developed, entire lobule is involved, and these small multiple microvesicular lipid droplets, they are accumulated together and they form the large clear macrovesicle, and we also call this the macrovesicular globules. And it can cause the compression and displacement of the nucleus to the periphery. Here in this figure, you can see macrovesicle as well as the microvesicle in the same hepatic globules. And here you can see the macrovesicle. Due to the macrovesicular steatosis, the nucleus is pushed to the periphery, that you can easily demonstrate in the higher view. Okay. Now fatty degeneration, which is seen in the cardiac muscle. Lipid is found in the cardiac muscle. The small amount of the lipid droplets, they are seen in the cardiac muscles. But fatty changes are developed when there is a hypoxia is developed to the heart muscle. If there is a prolonged moderate hypoxia occurs in the heart muscle, it due to the severe anemia is caused. It called the intracellular deposits of the fat. And due to the prolonged hypoxia, there is a alternate band of the normal myocardium with the fatty myocardium is developed, and it gives the effect of a alternate bands of the darker red brown and uninvolved myocardium, and we call this as a tigered effect. This image should be kept in your spotting exam. Okay, the tigered effect which is seen in the myocardium due to the prolonged moderate hypoxia. And the other pattern of the fatty changes which is observed in the myocardium is due to the severe hypoxia, and it is due to the diphtheриal infection or the myocarditis, and in which the whole myocardium, it undergoes fatty changes, and there is a no band of the normal myocardium seen in between the fatty myocardium. Okay. So this is the summary of the reversible cell injury morphological changes. First, I have told that it is the non-lethal injury with a retrogressive or the degenerative changes. The ultrastructural changes which are developed within the cell, they are demonstrable within the minutes to hour after the injury, and it is seen in the different organelles of the cell, and we can demonstrate it by the electron microscopy. And light microscopic and the gross changes are seen within the cells within the hours to day after the injury, and different morphological changes or the morphological forms of the reversible cell injury are the hydropic changes, hyaline changes, mucoid changes, and the fatty changes. So these are the morphological changes are seen in the reversible cell injury. Now we switch over to the morphological changes which are seen in the irreversible cell injury. I will share this MCQ later on with you. Now we coming to the irreversible cell injury. The learning objectives are same as I have discussed before, that we recall first the definition, then you can describe ultrastructural changes and explain the morphological patterns, as well as you can describe the gross and microscopic features with example of different organs. Okay. Now definition. When the continuous damage to the cells or the tissue by the injurious agent is occurs, it leads to irreversible type of injury, and at that time the cell cannot recover and it becomes dead or the dies, and we called it as the irreversible cell injury. What is the reversible cell injury? I have told that it is the mild form of injury and in which the injurious stimuli is removed and it leads to the mild functional as well as morphological changes within the cell. But this irreversible cell injury, it is the severe form of injury and it may lead to the cell death. There are two principal types of cell death which is seen in the irreversible cell injury, and that is the necrosis and apoptosis. Okay. Today I am going to discuss only the necrosis. Apoptosis I will not be discussing. Okay. So both these necrosis and apoptosis, they are differ in their morphology, in their mechanism of development, as well as by physiology, as well as in the form of disease development. So first we will see what is the necrosis. It is the morphological changes that follows the cell death in the living tissue or the organ, and it is occurs due to the progressive degenerative action of the enzymes on the lethally injured cells. Okay, so whenever severe injury to the cell occurs, there is a degenerative action of the proteolytic enzyme is started, and it leads to the cell death and irreversible cell injury. First, we will see what is the difference between necrosis and apoptosis. Apoptosis is also always pathological, and so there is always injurious stimuli is there in the development of necrosis. While apoptosis, it is the physiological or pathological. Why we are calling it physiological? Because it may also leads to the programmed cell death, and it leads to, so we also called it as the physiological. Necrosis, it is always followed by the inflammation, while apoptosis, in which the apoptotic cells, they are removed by the phagocytosis and it is not followed by the inflammation. Necrosis, it affects the clusters of cells or the groups of cells, while apoptosis, it affects the single cell. Okay, so these are the difference between the necrosis and apoptosis. Now we will see the ultrastructural changes which are seen in case of necrosis. These ultrastructural changes which are seen due to the denaturation of the protein which is caused by the intracellular acidosis, and it leads to the injury to the cell membrane, severe impairment of the phosphorylation of the cell, increase the permeability of the cell, influx of sodium and calcium ion within the cell, and decrease the intracellular activity of the cell. So these are the mechanism which is responsible for the development of ultrastructural changes within the cell due to the irreversible cell injury. Okay. Now, which changes are seen due to necrosis? The changes are seen in the mitochondria, in the nucleus, as well as within the cytoplasm. Now, the changes which are seen within the mitochondria, they are demonstrable by the electron microscopy. Okay. Now, which changes are seen in the mitochondria? The mitochondria becomes swollen. Same thing is happen in the reversible cell injury. But in reversible cell injury in the mitochondria, only what happened? The mitochondria becomes swollen and the amorphous density is developed within the mitochondrial parenchyma or the cell membrane. While here, in case of irreversible cell injury, there is a loss of intermitochondrial granules occurs, loss of its cristae and change in their shape, and sometimes it leads to the rupture of the outer mitochondrial membrane. And all these changes, they are demonstrable or seen by the electron microscopic examination. Now, changes in the nucleus. In the nucleus, the changes which we are easily seen by the light microscopic examination also. And these changes are seen in the form of pycnosis, karyorrhexis, and karyolysis. Now, what is the pycnosis? So here in this image, you can see this is the normal nucleus in which you can see there is a dispersed chromatin is also seen. But when there is a necrosis is occurs, the initial changes are seen within the nucleus in the form of pycnosis. It is characterized by the shrinkage of nucleus with increased basophilia and chromatin. It becomes condensed into the solid shrunken basophilic mass. Okay. If it becomes progress, then it leads to the karyorrhexis. Now, what happens in the karyorrhexis in which this pycnotic nuclei, it becomes fragmented and break up into the multiple small pieces or the fragments. And last is the karyolysis. This is the stage in which these fragmented or karyorrhectic nuclei, they undergo enzymatic digestion by the DNase and nucleus is completely disappeared and cell look ghost in appearance. Okay. So these are the nuclear changes which is observed within the cell at the time of necrosis. Then cytoplasmic changes, but here I can demonstrate the nuclear changes that are seen in case of liver cell necrosis. The first image in which you can see the normal liver cell, here dispersed chromatin is seen. If there is a necrosis is developed in initial stage, here you can see there is a clump chromatin and pycnotic nuclei is developed. Okay? And here in same image, you can also see the changes of karyorrhexis also. Here this is the karyorrhexis. Here karyorrhexis is more prominent, you can see. And the last stage, this karyorrhexis is disappear and these are the ghost cell without the nucleus and we called it as the karyolysis. Okay, so these are the different changes which is seen in the nucleus in case of necrosis. Now, changes in the cytoplasm. The cytoplasm, it looks more eosinophilic due to the loss of RNA and denaturation of the cytoplasmic proteins. So, cytoplasm, it looks more eosinophilic in appearance. Now, coming to the different morphological types of necrosis. These are the coagulative necrosis, liquefactive necrosis, caseous, fat, fibrinoid, and gangrenous necrosis. Now we can see one by one. First is the coagulative necrosis. It is the most common type of the necrosis in which the necrotic cell, it retains its cellular outline for several days after the injury. It is most commonly seen in the solid organ like kidney, heart, and the adrenal glands. It occurs due to the deficient blood supply or complete loss of blood supply or the anoxia. And the most common example of it is the myocardial infarction. Now what happened? In that, the injury, it leads to the denaturation of the structural protein, but it also leads to the denaturation of the enzymatic protein also. So as a result of which eosinophilic anucleate cells, they are persist for several days. Okay. And ultimately the necrotic cells, they are removed by the phagocytosis by the macrophages or the infiltrating leukocytes. And they can cause the digestion of the dead cells by the activity of the lysosomal enzymes of the leukocytes. Now, how they looks on the gross examination. When there is a myocardial infarction is developed, it is the cross section of the heart. This yellow area, that is the area of coagulative necrosis, and this pinkish or the red area, that is the normal myocardium. In the case of myocardial infarction, when the infarction is developed in initial stage, it becomes yellowish and soft in consistency. And as the disease is progress or the time passes, this yellowish area, it becomes shrunken and firm in consistency. Microscopic hallmark of the coagulative necrosis, it is the tombstone appearance of the cell. Now, what is the tombstone appearance of the cell? This is the microscopic figure of the kidney. Here you can see these are all the cells which looks ghost, in which the cytoplasmic border is preserved, but the cytoplasmic content are lost. Nucleus is also lost, and it gives the tombstone appearance of the cell. So this is the hallmark of coagulative necrosis. Now the second is the liquefactive necrosis. Now it is clear that coagulative necrosis occurs due to the loss of blood supply or due to the partial loss of blood supply. Now liquefactive necrosis, it is occurred due to ischemic injury or as well as due to the bacterial or the fungal infection. Okay, in which there is a degradation of the tissue is occurs by the action of powerful hydrolytic enzymes. And the most common example is the infarction of the brain or the abscess cavity. Now, whatever the pathogenesis is there, the liquefaction, it completely digests the dead cells, and as a result of which it leads to the formation of tissue into the liquid viscous mass. If the process was initiated by the acute inflammation, the material is creamy yellow in color and we called it as the pus. Okay. This is the gross example of the brain, in which here you can see the area of liquefactive necrosis. Here the abscess cavity is there, and center of this cavity it contains the necrotic material is formed. Now on microscopic examination, it contains the necrotic debris. Here you can demonstrate the necrotic debris. What is the necrotic debris? There is a structureless granular material. Okay, that is the necrotic debris. And here you can see some macrophages which engulf this dead or necrotic debris. Okay, and the cyst wall, it is formed by the proliferating capillaries, inflammatory cells, and the gliosis. Okay, and proliferating fibroblasts in case of the abscess cavity. The next is the caseous necrosis. So what happens in the liquefactive necrosis? In liquefactive necrosis, the tissue is transformed into the liquefactive mass, and it leads to the formation of dissolved mass walled off by the cystic cavity. Okay. Then caseous necrosis, it is a combination of both coagulative and the liquefactive necrosis. The term caseous, it means cheesy-like appearance, and it is derived from the friable yellow-white appearance of area of necrosis. The example of it is the center of the foci of tuberculous infection. This necrosis on gross appearance, it resembles like the dry cheese, and it is soft, granular, and yellowish in appearance. It is the cross section of the lung, and in which you can see this is the caseous necrotic area, and it appears like the dry, cheesy, and granular in appearance. And it is due to the histotoxic effect of the lipopolysaccharide present in the capsule of the tubercle bacilli. You have learned that the tubercle bacilli, in that there is an outer wall is made up of lipopolysaccharide. Okay, and this lipid material, it causes the histotoxic effect, and it gives the necrotic appearance, necrosis, and so this looks like a cheesy in appearance due to lipid histotoxic effect. The necrotic foci on microscopic examination, it looks like a structureless eosinophilic granular appearance, and the surrounding blue dots, they are the lymphoplasmacytic appearance, and here some giant cell formation is seen. And this is the whole is the tuberculous granuloma, and in which we can see the central area of caseous necrosis on microscopic examination, and that is the on gross examination appear like the cheesy necrotic in appearance. Caseous necrosis is also most commonly seen in the lymph node, and it is also give the same appearance as we have seen in the lung. Then the fat necrosis, it refers to the focal area of the fat destruction. The classical example of it is the acute pancreatic necrosis and traumatic fat necrosis, which is seen in the breast. Now, in acute pancreatitis, what happens? The pancreatic enzymes, they leaked out from the acinar cells and the duct, and they can cause the liquefaction of the membrane of fat cells into the peritoneum, and as a result of which the lipase, it splits the triglyceride ester which is contained within the fat cell into the fatty acids, and when it combined with the calcium, it produces the grossly visible chalky white areas. We also call it as the saponification of the fat. So whenever there is an acute pancreatitis is developed on the surface of pancreas, these chalky white deposits are seen, and that is called as the fat necrosis or the fat saponification. Okay? And when we see on the microscopic examination, these necrous fat cells, they give the cloudy appearance. Here, they are the necrotic fat cells, and it gives a cloudy appearance. And if it is of long standing, it gives the amorphous granular appearance. Here you can see the amorphous granular appearance of the basophilic material. It is seen the acute pancreatitis. And calcium soap identified by basophilic material deposition. This is the calcification. If these chalky streaks deposited in the cells for longer duration of time, then it leads to the formation of the calcium soap. Now, what is the mechanism of it? Development of calcium soap. If it is of long duration, it leads to the dystrophic calcification, and it leads to the formation of calcium soap. Then next is the fibrinoid necrosis. It is a special form of necrosis which is seen in the immune reaction. Whenever there is an immunological reaction which involves the blood vessels, and as a result of which there is an antigen and antibody complexes, they are deposited in the wall of the arteries. And we also called it as the immune complex mediated reaction, and in which it leads to the pink amorphous appearance in H-stained sections, and we called it as the fibrinoid-like material, and so we called it as the fibrinoid necrosis. The example are the immune complex vasculitis and the autoimmune disease. Here you can see this is the blood vessel wall. In the blood vessel wall, there is a fibrinoid necrosis is seen in the form of granular eosinophilic material, and it is developed due to the deposition of immune complexes or the antigen antibody complexes. And in the surrounding area, you can see there is a accumulation or the deposition of acute inflammatory cells, and that is the polymorphs or the neutrophils. And here and we called it as the leukocytoclasis. Okay, and in the center you can see the hemorrhage. So this is a fibrinoid necrosis which is seen in the immune mediated reaction. So what is the outcome of necrosis? Any necrosis in which the last outcome is this: most of the necrotic cells and their contents, they are disappear due to enzymatic digestion or the phagocytosis of the debris by the leukocytes. If necrotic cells at the cellular debris, they are not promptly destroyed or reabsorbed, if they are very large, then they provide the nidus for the deposition of calcium salts and other minerals, and it leads to development of the calcifications. And so this area become calcified, and this phenomenon we called it as the dystrophic calcification. Okay. So in summary of necrosis, with continuous damage, the injury become irreversible, and at that time the cell did not get the time to recover and it becomes dies. Okay. And ultrastructural changes are seen in the form of increased eosinophilia, nuclear shrinkage, fragmentation and dissolution, that is the karyolysis. breakdown of plasma membrane, organellar membrane with leakage and enzymatic digestion of cellular contents. And then example of different necrosis, that is a coagulative, caseous, liquefactive, fat necrosis and the fibrinoid necrosis. So these all about the morphological changes that you can see in the reversible as well as reversible cell injury. In reversible cell injury, mainly in case of a necrosis. Apoptosis, it will be taken in the second lecture. Okay. So if you have any query, you can share with me. I will give 2 minutes to you.